We are delighted to bring you this seminar presented by LabRoots and in collaboration with the National Institute of Health. Welcome to our track on the NIH Brain Initiative, a multidisciplinary approach to neuroscience. We're glad you can join us today for this panel presentation titled Anatomical Characterization of Neuron Cell Types in the Mouse Brain. It is my pleasure to introduce a panel of experts who will be speaking on the topic of resource and tool development. For a full listing, please click on the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speakers will respond to you back via email. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming our panel of experts.
Hello, everyone. My name is Hongwei Dong, Professor of Neurology at USC. Today, I and two of my collaborators, Dr. Georgia Ascoli and Bianca Lim, talk about our U1 grant that is funded by Brain Initiative. So the goal for this project is to identify different cell types in the mouse brain and understand each individual cell types, their specific functions, and eventually understand how these different cell types, they contribute to the neurological disease and the neuropsychiatric disease. In fact, Romani Cajal already laid the ground foundation for this kind of work. As you can see in his beautiful drawings, he showed in the retina has several different neuronal cell types with different morphology, and they send the projections to the tectum. In the tectum also has different cell types, display different neuronal morphology. In fact, in the last several decades, and the whole neuroanatomy field is trying to do this work on refine the anatomy, identify different cell types, understand their local circuitries and their long-range projections. So for our work also is the same thing. We focus on several different aspects. We systematically map long-range connections from different brain regions. And in each individual brain regions, we try to catalog different cell types based on their neuronal morphology and also define their cell type specific neurocircuitries. All of this work eventually will be hopefully will lead to our ultimate goal to understand how these different neuronal cell types and also single neurons, their input-output organizational principles. And for today, our talk, I'm going to focus on the first part to report our progress of construct a whole brain wiring diagram. And how these different neurocircuitries associated with individual brain structures, they put together as neural networks for conduct different behaviors and functions. And I'm going to also lay the framework for general strategies, our grant, to how we systematically catalog neuronal cell types. And Dr. Georgia Ascoli is going to talk in details how we define cell types based on their neuromorphology, and also another project called the Shotgun Project. And Dr. Lim is going to present our effort of developing cutting-edge technologies to define the cell type-specific neurocircuitries. So for ultimately what we are really trying to understand is, so if the individual neurons are building blocks of the brain, and how these individual neurons, they are organized together to conduct a sophisticated behavior. This exactly follows the same principles, just like individual piano keys. When they have different combinations, they can play beautiful music piece. So a little bit of background about our mouse connectome project that was launched about 10 years ago from my lab. And this is one of the first Brain Initiative funded project. The goal for this project is construct the whole brain 3D connectivity atlas. And here's one representative images to show we use different fluorescent tracers to label the different pathways. As you can see here, red color fibers and their terminals, and green color axons and their terminals. At the same time, we also label the upstream neurons, as we show here, the pink dots. And so the challenge part for us really is in the mammalian brain has about 800 to 1,000 well-defined brain structures. How we can systematically, structure by structure, map their neural circuitries and then put them together as neural networks. So about 10 years ago, we started from first look at the neural circuitries in the cortex. The whole cortex has about 60 different cortical areas. We started looking at their circuitries for each individual cortical areas, and then eventually we put together as this published work called Neural Networks of the Mouse Neocortex. And this work, in fact, set up the entire framework for us to understand the whole central nervous system, neural circuitries, how they organized. So a little bit of background just in terms of our techniques. So we use this called tractor tracing method to do the stereotaxic injections, inject the tracers into the mouse brain. For each injection, we have a mixture of one anterograde tracers. As we show here, the green color axons come from the injection site. This is labeled as MOS, secondary motor cortex. These axons project to the contralateral side, their terminals. At the same time, in this injection site has a mixture of retrograde tracers, which can be picked up by axonal terminals by their upstream neurons and retrogradely back label these neurons. As we can show here, this pink dots, that's the upstream neurons. They project back to the injection site. So we do this, use these systematic injections, injecting into the entire cortex. It's about 300 injections, 600 pathways. And then eventually we put together and construct this, the most detailed connectivity map of the entire cortical connections. And what we find out is, in fact, the entire cortical, all the different cortical areas, they are organized as a modular subnetworks. For example, entire somatic sensory motor areas include primary sensory, primary motor, secondary motor, secondary somatosensory. They are organized into the several distinctive neural networks in conduct different behaviors. For example, this blue color labeled pathways, they are organized by several individual areas. They are fully connected with each other in the control of our facial movements. And this green color labeled pathways, they're organized by several areas. They're also fully connected with each other, conduct and control the upper limb movements. And the red color pathways control the lower limb pathways. So these modular subnetworks allow the sensory and motor areas directly interact with each other and conduct different sensory motor functions. So in contrast, so this visual area and auditory areas, they are heavily connected with several other areas called high order association areas. They are distributed along the media bank of the cortex. These pathways we put together as called media cortical networks. They are processed and transmit this visual, auditory, spatial sensories to the prefrontal cortex. Another side of networks, that is the cortical areas, they are distributed along the lateral edges of the cortex, include insular cortical areas, temporal association area, and also the internal cortical areas. They are organized as so-called the lateral cortical networks, and these networks, they are processed visceral, gustatory, olfactory information and also function related to self-awareness. So put together, we look at the entire mouse cortex. They're organized into this about 10 different subnetworks. They conduct the different functions and put all together, all these different individual subnetworks. They are convergent onto the prefrontal cortex, and the somatic sensory subnetworks, they are convergent onto the dorsolateral prefrontal areas, and the medial and the lateral cortical areas, they are converged on the ventral medial cortical areas. If we look at it on the flat map of the mouse brain, we can see the entire cortical areas. They organize into the different subnetworks. They circulate throughout the entire cortical hemisphere. So what happened after we mapped the entire cortical networks? The next step is we're trying to understand individual cortical areas and also their subnetworks, their projections to different subcortical structures. So the first structure we're looking at is the striatum, because the striatum receives all individual cortical areas. They generate projections to the striatum, and the striatum play important functions in the control of the motor, cognition, and other important functions. This also is published work, and we put together as this the mouse cortical striatal projectome. And we mapped individual cortical areas and generate this very detailed cortical striatal projection map. And then, as you can see here, each individual cortical areas, they project to the striatum, they all display distinctive projection patterns. And then we did the quantification, and we did a network analysis, and then we look at from how these different cortical areas and also their different subnetworks, how they project the striatum. Based on this, their projection patterns, we basically subdivide the entire striatum into these 11 relatively larger communities and 29 smaller functional domains. And when we look at how they work, we find out actually the cortical areas, they are organized as subnetworks. They display overlap projection patterns in the striatum. For example, what we show here is the entire somatic sensory motor cortex. Remember, they are organized as 4 parallel pathways in control orificial, upper limb, lower limb, trunk functions. And in each subnetwork has these somatosensory, somatomotor areas. They heavily connect with each other in the cortical areas. And then their projection terminals in the striatum, they are overlapped. So from this map, what we can see here is the trunk area subnetworks, they project to the relatively dorsal parts, and then is lower limb, then is upper limb, and then all the orificial related cortical areas, they project to ventrolateral side of the striatum. Put together, they form this kind of upside down homunculus projection patterns. And then the medial cortical networks, these different cortical areas, they project mostly to the dorsal medial striatum. Cortical areas within the lateral subnetworks, they project to ventral medial side of the striatum. But however, what we really should pay attention is each individual cortical areas, their projection pattern in the striatum, they are very unique. But each individual striatum domains, they receive convergent inputs from a unique subset of cortical areas. Put all together, we generate this hypothesis functional model of the dorsal striatum. So basically, the 29 functional domains, they receive unique different cortical inputs. For example, this here we show in the middle of the stratum, next to the lateral ventricle, this blue color vertical stripes. It receives specific input from primary visual cortex, internal cortex, which speaks their function in the visual spatial integration. And the ventrolateral side of the stratum receive specific input from all the orofacial related somatic sensory motor cortical areas. And after we look at the entire cortical striatal projections, obviously the next natural question is how these different individual stratum domains, what is their downstream projection patterns? So we then made injections into all these individual functional domains of stratum and look at how the stratum project its downstream structures, pallidum, nigra, and then project back to the thalamus. Hopefully we can finish this work in a short amount of time. So the next question really is asked, how about cortical project other subcortical structures? For example, here's the superior colliculus. And for this part of our work, we also generate called the mouse cortical tactile projectome. And that's also analyzed many injections in the cortical areas. And then we put together this paper. This is unpublished data. We used about 86 cortical tactile projection pathways. And the same thing, we look at each individual cortical areas, their projections to the stratum. And accordingly, based on these connectivity patterns, we subdivided the superior colliculus into 4 different columns. And as we can show here, the different columns receive the convergent inputs from different cortical areas. For example, the lateral side of this column receive all the inputs from somatic sensory motor areas, and the medial column receive inputs from visual, anterior cingulate, and other cortical inputs related to the visual and eye movements. And then we make injections into this superior colliculus and then put together this wiring diagram of the entire superior colliculus. And so this is our general strategy, step by step. And then eventually we're going to map the connectivity for the entire brain, and also look at how the different circuitries put together as global neural networks. So, and another really important set of the cortex is this called the limbic system, classic limbic system. They are organized by hippocampus, amygdala, and medial prefrontal cortex. Their projection patterns mostly to the hypothalamus and in the control of this emotional related behavior. So for this part, we also made a good progress. We put together of this hippocampal connectome paper. And for this paper, a little bit different is we also look at how the connectivity relate to the gene expression patterns. So the general strategy is we systematically analyzed about 2,000 genes from Allen Brain Atlas database. Look at in each individual component of a hippocampus. For example, CE2 here, which genes expressed in the CE2 and which genes into the different segments of the CE3, CE1 speculum. And then based on these gene expression patterns, we subdivide each individual hippocampal component into the different molecular domains. After that, and then we examined each molecular domains, what's their input, output, and then put together this 3D atlas of the hippocampus and also hippocampal wiring diagram. As we show here, each individual molecular domains, they display unique patterns of the gene expression, but also they correlate with the neurons. They send projections to different projection targets. And once we put together this map, and we also look at each individual brain individual hippocampal molecular domains, what is their global projection patterns. For example, like here showed different neuronal populations in the internal cortex, they project to different components of this speculum, and also their projections to the mammillary body. And then here together, we put this hippocampal wiring diagram. This one you can find from our website. So, we put together this global wiring diagram that will help us to really refine the anatomy and then identify in each individual brain structures with these smaller subdivisions, and then further subdivide, look at each individual small subdivisions, how many different cell types there, and then look at how these different cell types, what is their unique connectivity patterns that will help us eventually understand their unique behavior and functions. And so after we put together this wiring diagram for each individual brain structures, we look at their axonal projections, and then we start use this called projection defined cell types, and then to catalog this neuronal populations into different subpopulations based on their connectivity. Just like what I showed here, this is a region, one individual region, they project to multiple targets, and in each individual regions, they have these different cell types. And then we make injections of retrograde tracers into their different cell types. And then they back label neurons in their regions. And then we look at which neurons project one target or two targets or different targets. So the whole projection pattern, the strategies that we use anterograde tracer, look at their projection terminals, and then use retrograde tracers, define the different neuronal populations. And then we carefully look at their anatomical distributions, and then look at their morphology, and then characterize their cell type specific projection patterns. As what I showed here, that is, we define the hippocampal's different molecular domains, and then make injections of these retrograde tracers into their different targets. For example, in the last sections here, yellow color, that is injection retrograde tracers into the thalamus, back labels the yellow neurons. They're distributed in the deep layers of speculum. They all thalamic projection neurons. And this cyan these pink neurons, they project to the median mammillary body, and this thin layer green color labeled neurons, they project to lateral mammillary nucleus. And then we look at characterize how these neurons, they project one target or different targets, and then we do the quantification. We characterize their different population based on their projection targets. And then we can match them with their molecular domains. So this is the ongoing work, and hopefully in the future, we can also use a different approach to look at exactly the same neurons and look at same neurons, which molecular markers they are expressed. And obviously, after we look at the individual neurons, where they project to, and we use the rabies labeling, we can also reveal their detailed neuronal morphology. As what we can show here, these green color neurons, they project to anterior ventral thalamic nucleus. You can see this neuron display extremely complex dendritic morphologies, which indicate how the information can be processed in these neurons. And also we can map the axons from these neurons. And these different colors represent different neurons. So this red color neurons project to reunion thalamic nucleus, and the pink neurons here project to retrosplenic areas. And also we use this same strategy, label different neuron populations, also look at in the other different brain structures, such as superior colliculus. We have developed neuroinformatics tools to characterize their neuronal morphology and also catalog the different neuronal populations. And Dr. Giorgio Ascolini is going to give more detailed talk about how we characterize neurons based on their morphology. And also Dr. Lim is going to talk about how we characterize the cell types and then define their cell type specific neural circuitries. And also we have collaborations with other groups funded by Brain Initiative, look at their molecular signatures and also physiology functional properties. This work, we have well-developed collaborations through our U1 grant and also with other groups funded by Brain Initiatives. And finally, I want to acknowledge my lab. Without them, this work cannot be done in such scale. And also, I really appreciate my the whole group and also my collaborators, Giorgio Ascoli, Bianca Lim, in Wickensham, and also other collaborators. We are working together on other projects also funded by Brain grants.
Hello, my name is Giorgio Ascoli. I'm a professor at George Mason University in Fairfax, Virginia. And today I'm going to follow through the tale of what Hongwei Peng described to talk about two projects. One is a database of neuron reconstructions that is very much inspired by the need to catalog all neuron types, in this case based on morphology. And the other is a very novel way of analyzing projection types based on retrograde track tracing, which was introduced in Hongwei's talk. So the importance of morphology in neuroscience has been understood for now over a century since the seminal work of Ramon Cajal. And much of the research today asks questions of what happens to morphology when one analyzes similar neurons in different species or different developmental stages or upon manipulation that can be due to biological injuries or pharmacological interventions. Although there are neurons that are different because they belong to different types, we always have to remember that in biology, no two objects are exactly the same. And so there is an important question of asking whether neurons and their shapes belong to a continuum or they form distinct classes. And to paraphrase Orwell's Animals Farm, I would say that each neuron is different, but some neurons are more different than others. Mathematicians have come up with methods to understand groupings of objects that are similar but not identical. And one of these methods is cluster analysis. The idea is to represent parameters of the shape, for example, the total length of the arbor or the number of branches or the volume invaded by the branching structure, and plot those in a multidimensional space. And because every neuron is different, no two neurons will occupy the exact same position in space, but they will tend to form clusters. Those are what correspond to morphology. So in order to do this in large scale and with very systematic approaches, again, since the early days of neuroscience, Cajal had already defined the process for reconstructing the shape of neurons with three major steps, which are labeling, visualization, and tracing. And Cajal did that mostly with bulk labeling and specifically Golgi staining, optical wide field microscopy, and then hand tracing, pencil on paper. Since then, the community has developed a variety of sophisticated and specific methods for labeling or visualization, and tracing has been paired to computerized stations that allow the three-dimensional representation of each and every position of tracing point in space, so that the arbor is represented as a series of vectors. Very much the protein can be represented in structure as a series of dimensional coordinates. In the case of neurons, these series of three-dimensional coordinates are connected in arbor. So many years ago, in 2006, we launched a resource called neuromorpho.org to host any and all digital reconstructions of neuronal and now glia morphology collected by hundreds of different laboratories worldwide from any animal species, brain region, mental condition, and cell type. And at this point, in 2020, we've passed over 120,000 neurons, and these neurons are downloaded in a large variety of labs for great diversity studies. And these reconstructions have quite a bit of detail in their branching structure, but the more impressive feature of the database is, in fact, its growing size that truly allows for integrative studies that would not be possible in individual. So in the latest version that was released last September, there are about 120,000 neurons from 650 labs. 40 million tracings have been downloaded from pretty much countries listed in the United Nations. These are from not only the most common model species, rats, mice, and flies, but pretty much any phyla from T. elegans to humans. And this is based on a literature, continuous literature search, which now contains over 43,000 references. And we're now going towards 2,000 studies published based on data or describing data from neuromorpho.org. And now there are also hundreds of NIH grants that are funded based on data from neuromorpho.org from 255 PIs, many universities, and hundreds of millions of dollars. So mostly the reconstructions of the neurons are used for modeling, for comparative morphometric analysis, for studies of potential connectivity, and for visualization and educational purposes. Since the beginning, however, the applications in academia and in biomedical research were not the only ones for data. For many years, these data have been used for a variety of STEM and arts applications, including applied math Olympiads, neuroscience for kids, variety of artistic renderings, including audiovisual representations for a play on Wittgenstein, and many more. And one of the most recent applications is in aiding the cell sensors for the brain initiative cell sensor. So the database is maintained through continuous pipeline of process that starts from searching the literature to identify data, and we have an ongoing continuous database literature that is continuously annotated with a combination of language processing, automated parsing, and custom notation. In the same data search, we also analyze reports of the usage of the data, so we can then return to the authors and to the funding agencies with updates on the usage of the resource. We then request the data. At the beginning, a minority of the data were deposited in the database. Now, approximately 80% of our requests are honored by the authors. So we receive the data and we process those data to standardize them into an open, non-proprietary format. But we also check for common inconsistencies in data files that might affect downstream analysis. We also annotate the metadata from the data set that we receive, linking them to evidence in the literature, and then we update the schema of the database to make sure that all data are searchable by metadata as well as by morphology. Finally, we release the data first on a review site so that we allow authors and our technical staff to check that the data are complete, and finally the public when we then release announcement to the community. And all the data that we release is open source, open access, so it's completely free to the users, and it's mirrorable with no limit. So the literature itself is also searchable through neuromorpho.org by status. It's important to know that because the mass of the data are increases quasi-exponentially, it means that most of the data is from the most recent time. And not only that, but the complexity of the data is also changing and increasing over time. So most of the data from the early days of the database were from dendritic reconstruction from slides. And some of the data from recent times, including from the ICN project, include both dendritic and full axonal reconstructions, which constitute most of the cabling. So in the second part of my talk, I would like to talk about a method to analyze retrograde track tracing data in order to classify axonal projections in a way that is complementary to the brute force neural reconstruction. And the idea here is that every brain region, especially every cortical brain region in mammalian brains, projects multiple targets. And this can be viewed from microscopic regional connectivity levels. So if a region A projects to a target region B, it means that there is at least one neuron type with a soma located in A whose axon extends in B. And basically, depending on how one parses anatomy of the brain, in how many parcels and how many regions, the number of the target regions for a given source region will change. But for the cortex or mainstream atlases, this number is typically between 10 and over 30. And these are examples of the resources from which some of these data are taken, including the iconic film that Hongwei mentioned. So what kind of axonal architecture at the single neuron level can subserve microscopically the macrostate of these regional connections? So there could be one neuron type for each target region. So every neuron type might project to a single region, and M target regions, there might be M neuron types. One could also envision a single neuron type that routes axons connecting to all M target regions. And of course, there are many in-between cases that, for those who have studied these projections, are much more realistic, which is an intermediate number of neuron types that will project to a subset of the target area. And our point of view is that if two neurons project to two different set areas, they represent different classes that will affect the network in different ways. So this is a form of neuronal classification. Now, it might be simple or too simple to classify neurons purely based on their axonal projection target, but it's really not very simple. Even if one simplifies the entire human brain structure in simply top, bottom, right, left, front, back, or if you like, four lobes, or bilateral right and left structure. So this is the equivalent of approximating the whole brain to a 2 by 2 by 2 cube. Now consider now an axonal projection. It will be simply a binary vector of 8 elements. And so we can compare two neurons as two vectors of 8 elements each. And these vectors are binary, where 0 means that a given neuron does not project to a given source, to a given target, and 1 means that the same neuron projects. So in this case, these are two binary vectors that are not the same, represent two neurons that project to two different subsets, this 2 by 2 by 2. So even in this very simple representation, the number of possible neuron types you count for sublocation is 8 times 2 to the 8 minus 1, or 2,040 different neuron types, just out of a 2 by 2 by 2 cube. Now, if one counts the approximately 1,000 different brain regions that are atlas in modern anatomical parcellation, and one does the same calculation, this would obtain a number of neuron types that is far greater than the total number of neurons in any and all species of all animals in the entire known universe. And the curve is an exponential. So the truth is that not all possible classes exist for a given part, for a given source and a given brain, and instead only a subset of classes is typically found in any one region. So for example, if we look at a critical region projecting to 7 target regions, perhaps only 20 of the 127 neuron classes. And knowing which classes are present, which classes do not exist, not only that, but also how many neurons there are in each class, that's basically the sense of challenge for anatomical projection. So how can we solve that anatomical challenge? And that there are many institutes and agencies especially interested in this, including the US Brain Initiative, the European Human Brain Project, and also massive large-scale industrial initiatives, including at the Allen Institute, at Janidia for mouse light, and the Brains Math. So one possibility is to reconstruct all the neurons and the external projection. And this is something that is very time-consuming and labor-intensive by great progress on automation. However, this process is not very scalable, because even if we were to reconstruct only a representative sample, say a dozen or so cells for each class, this would in fact demand an impractical number of constructions. And you would probably still miss the rarest type, the neuron projection type, for which there are fewest. Not only that, but this operation would then need to be repeated for every source region in the brain. And so if one is interested in the entirety of the brain, with say 1,000 source regions, this would require multiplication of 1,000 in experiments. So as an alternative, we can think of a thought experiment based on Hongwei's beautifully made retrograde projection, which is suppose that we can inject an arbitrary number of different colors retrogradely in all the regions of the brain. Let's color those C1 to CM for the M target region. And then what we can do is simply go to the source region and count the number of cells that are colored with each unique combination of colors. And if that were doable, we could even do that automatically in a very expedient way, and we would be able to determine the number of neurons with each projection. However, this thought experiment has two major limitations. One is that in practice, we can only do multiple retrograde projections to a limited number of regions. And the second is that when we inject retrogradely a label, we only hit part of the target region. In other words, we can only label a fraction of the existing fibers and not 100% of the fibers. So we're bound to miss of the cell in the source. So shotgun projectomics is a way to tackle these challenges. And to explain how that works, we can start from the simplest case, which is a region projected 4 target regions, n equals 4. And this only has 15 projection types. And because there are only 15, I was able to manually draw them all in PowerPoint. And so these are color-coded. Say type 1 only projects to region 1, type 15 projects to all 4 target regions. And so let's now imagine that we only have 3 retrograde colors and not 4. So we can design 4 experiments. In each experiment, we will leave out one of the target regions. So let's call the 3 colors green, red, and blue. And let's remember that when we hit a target, we will not hit 100% of the target, but we'll only hit a fraction of it. So for every experiment, we will run then 4 experiments. We'll have a combination of 3 colors, and the possible combinations of 3 colors give rise to 7 different counts. Only green, only red, only blue, red and green, red and blue, green and blue, red, green, and blue. So which of these 15 counts contribute, which of the 15 neuron types contribute to the count of the neurons that are just color green? Well, for sure, all the cells that are green must project to target count 1, otherwise they would not be green. However, remember that not all the neurons that project to target 1 are green because we're only hitting a fraction of target 1. Let's say K1 is that fraction. So we can say G equals K1 times T1, where T1 is the count of the cells that project to target 1. But that's not enough because we're not putting any color in target 4. So all the neuron types project to both target 1 and to target 4 also going to be green. So we have to add also K1 times T1, 4, which is the number of projection types that hits both target 1 and target 4. But that's also not enough because we also have neurons that project 1 and 2, but they are in the penumbra of 2, the fraction of the cells that invade 2 that are not picking up the red color. And now you get the drill. We do the same for 1 and 3, for 1, 2, and 3, where both 2 and 3 are in the penumbra, and all of the above that also hit 4, for which we are completely blind. So we can write this very complex equation. And for each of these 4 experiments, we can similarly write 7 such equations, 1 equation for each of the compilations of color. So we have 4 experiments, 7 equations for each experiment, which makes 28 equations. Now, how many unknowns do we have? Well, we have 15 unknowns, which are the numbers of neurons in each type. And then we also have the fractions of colors that are picked up, those K1, K2, K3 parameters. We have 3 of those unknowns for each of the 4 experiments, so 12 unknowns. So we have a grand total of 27 unknowns and 28 equations. So we can put this system with 28 equations and 27 unknowns in a numerical optimizer and let the computer crank the numbers and find possible solutions. So we did computational analysis of this using evolutionary algorithm and surrogate data. And these data suggest that, in fact, we can reconstruct correct pattern of data robustly in a variety of different scenarios and with minimal error, meaning error below 5% on the count, and with a limited number of experiments, approximately 5 experiments per condition. So we're now starting to analyze data from the Dong lab, where they in fact are able to do triple retrograde injections in a very routine way, but also quadruple injections with some effort. And they're now testing and piloting the ability of doing quintuple injections. So this is now a very exciting time because we might soon be able to have actual numbers of counts for prototypes based on this. And just as a reminder, because things grow exponentially, the system is quite powerful in the sense that with quadruple injections, which again are doable with current technology, we could resolve fairly easily projections for up to 12 regions. Now remember, the number of neuron types, 12 regions, is of the order of 2 to the 12, which is approximately 4,000 different neurons. So a staggering number of neurons. The most important thing of this technique is that it scales very well, because once you're counting neurons and you're slicing brains, and in fact, look at not a single source, but any of the sources that are projecting to the same combinations of targets. So especially in the cortex, this might allow you to resolve the neuronal census problem, multiple sources with the same set of. So I'm going to stop here and acknowledge the sources of funding, mostly NIH, from both the Brain Initiative, the BISTI program, and the CRCNS, Collaborative Research in Computational Neuroscience program, as well as philanthropic foundation, philanthropic support from Melton Foundation and private support from Northrop Grumman Corporation, as well as a variety of past funding from government agencies, including NIH, the National Science Foundation, and DOE, such as the Office of Naval Research, Air Force, and DARPA. And last but not least, I would like to acknowledge the contribution of fantastic members of the center here at George Mason, specifically for the project that I highlighted in this presentation. And Linguist and Dick Wheeler are the leaders of neuromorpho.org and of the network analysis in my lab. And among the PhD students, Masood Arkram and Patricia Maraver, Kaivan Baijari are very much fueling the progress in neuromorpho.org, and Siva Venkatesh and all the evolutionary algorithms to prove the feasibility of I'll stop here, but all the data are available online, and I'm also available for questions at this email address.
Hi, my name is Byung-Guk Lim from UCSD. Thank you very much for this opportunity. You already heard from Hongwei and Giorgio about the large-scale analysis of the neuronal connectivity. So my talk is a little different. Mine is more focused on the specific neural circuitry. So as you know, the brain is the connection of the many different cell types. So I mean the many different cell types, meaning that there are tons of different cell types connecting to the different cell type. We don't really know how they are actually different. And you know, other problem is not just cell type, it's connection. It means that how the different cell type connected to what type of cell type is actually really puzzling. And without fully understanding the cell type specific connection, it may be really difficult to understand how brains send information together. So connection, you know, in brain, connection is synapse. Each neuron has more than 1,000 synapses, meaning that connect to 1,000 different targets. And then number and the specificity of the synaptic connection is very important, and especially the price we controlled during the development. And the change in the synaptic connection, meaning that strength of the synaptic connection change, is actually the fundamental unit of the information processing, underlying memory, and other decision making. So it is very important to understand the connection of different cell type in like synapse specific manner. So how I talk about the actually most amazing work that actually tried to identify the projection of individual cell in the cortex and the other brain area. But at this moment, all the information we're having is actually where those neurons project to. So they are actually looking at the axonal fiber. So I thought that we need to have a tool to differentiate the axon fiber and synaptic baton in the same neuron to clearly understand the anatomical organization. So another problem is the brain is actually very heterogeneous, even in the same brain area. So when you look at the right panel, here I labeled the primary cortex neuron projecting to dorsal medial stratum with the green by injecting retrograde virus expressing green fluorescence protein in the dorsal medial stratum. And then I labeled the neuron projecting to dorsal lateral stratum with the red fluorescence. So when you look at the primary motor cortex, there are not much of segregation. There is, but more likely they're intermingled in the similar brain area. So it's a little difficult to differentiate the neuronal population based on the projection. So a lot of effort has been done and still going on to generate a transgenic animal expressing Cre recombinase under the specific promoter, which marking the specific cell type in cortex. So there are a lot of list of the transgenic animal expressing Cre and marking the different cell type, but still, each same cell neuron, same type neuron labeled by this Cre line may actually project to the same brain area. So we need to find a tool to define the cell type and projection specific manner. So this is very daunting, so meaning that you have to have a specific tool, especially the viral tool, to label the specific cell type and specific projection. So to understand, to apply this tool, we actually study the basal ganglia. So my lab has been studying the role of the basal ganglia in the movement disorder and the decision making. So this slide is actually the very classical diagram of the basal ganglia from stratum, globus pallidus, and thalamus and cortex. So I guess this diagram has been used for more than 20 years. But while I'm keep studying the role of the basal ganglia, I'm beginning to realize that the diagram and circuit organization is not as simple as this diagram. Especially their synaptic connection is very difficult, very actually different than actually proposed. So in this, presentation, we are focusing on the globus pallidus. So globus pallidus is covered in neuronal population. Actually, more than 60% of the neurons are pop-almin positive. So using the pop-almin Cre animal, which can define the globus pallidus neuron expressing pop-almin as a cellular marker, we developed the virus tool expressing red fluorescence protein to mark the cell body and the axon fiber, and the synaptophysin tag, the GFP, which can mark the presynaptic terminal specifically. So we inject this virus, which expressing this protein and Cre-dependent manner into the globus pallidus of pop-almin Cre animal. And then we found that actually, yes, they are projecting to many different targets. Conventionally, they are projecting, conventional diagram only talking about the subthalamic nucleus, STN, but we found many different areas projected by this pop-almin positive neuron in the globus pallidus. So what we know when we look at the little deep structure, yes, each globus pallidus neuron make a synapse onto each other's. And then we are able to differentiate the axon fiber and the synaptic puncta based on the green puncta and the red fluorescence protein. So when you go to the other brain area, it becomes more obvious that STN, this is the thalamic nucleus, their fiber and the synaptic connection can be clearly differentiated. And when you look at the SNR area, you can even look at the cell shape of the postsynaptic cell just by looking at the just presynaptic terminal made by the globus pallidus pavlov neuron. And then this is a whole section confocal image showing that just by looking at the axon fiber, it may be really difficult to understand that their actual connection by single neuron. And then it did become more obvious when you do the 3D structure. So here we show that the same cell type, genetically defined the same cell type, pavlov and positive neuron, globus pallidus, project to many different targets, right? So this actually we were able to achieve with this specific viral tool expressing red fluorescence protein in the cell body and axon terminal, and the green puncta at the synaptic terminal. So we know that this neuron, genetically defined the same population in globus pallidus project many different targets, but we have a very specific question. Okay, it's possible the same neuron project to many different targets, and different population neuron project to the different target. To understand the function of the globus pallidus overall the basal ganglia, and identifying these two possibilities and differentiating these two possibilities is very important. The question is, how can we differentiate this genetically defined two different populations may project to the different target? So we engineered the virus. Actually, we engineered the rabies virus. So you know that a rabies virus is a very potent retrograde tracer, and Hongwei already showed the many beautiful data that are labeled by the rabies virus. It's important for the retrograde infection, and they express the transgene very high level, but it's induced toxicity, so you cannot use for a long period of time. So property of the retrograde transduction is mainly because of the glycoprotein on the surface of the rabies virus. So using that idea, we actually pseudotype the lentivirus from horse, equine infectious anemia virus, EIAV. So actually pseudotype this, EIAV virus with the glycoprotein of the rabies. So this virus can go retrogradely by infecting the axon terminal. And then it has a DNA genome in the viral particle, so it can generate the stable expression of the transgene, and it doesn't kill the cell. So you can do long-term study with this viral tool. So using this idea, we actually further modify the genome of the virus. So we actually using the flip recombinase and the decrease recombinase combination. So we generate the retrograde EIAV virus expressing flip recombinase in pre-dependent manner. So when this virus is injected into the target area, it will be retrogradely transduced to the presynaptic cell. Then only the cell expressing CRE will express flip. And then when we inject the AAV virus expressing transgene in flip-dependent manner, so only the neuron projecting this target will actually express this transgene. So when we inject this virus combination, so when we inject the retrograde EIAV expressing flip recombinase in pre-dependent manner into the SNR, and then injecting AAV expressing GFP in flip-dependent manner into the globus pallidus, the pavlov-Cree animal. And then to differentiate the other population, we inject this same approach, the retrograde EAAV expressing flip recombinase in pre-dependent manner into the paravascular nucleus, and then injecting AAV expressing GFP in flip-dependent manner into the globus pallidus of the pavlov-Cree animal. When we do that, we found that the neuron projecting to SNR, doesn't go to the paravascular nucleus. And then we also found that globus pallidus pavlov neuron projecting to paravascular nucleus doesn't go to the SNR or STN. So as you can see in the first panel, actually the location of the cell projecting different target are actually totally different even within the globus pallidus. This gives you the clear idea that even though genetically the same population based on where they project to, they can be totally different circuitry. But this one is actually looking at the group, looking at the group of the neuron. So we actually thought that to fully understand the anatomical organization, they're actually their projection or the organization of the individual neuron is very important to be examined. So to get the single neuron tracing, we actually applied a new technique. So to get the single neuron tracing, we need to get the high resolution image, and then also they have to detect the synaptic bouton, and we have to get the reliable fluorescence labelings because if you lose the fluorescence, you will not trace the neuron. And then it's better to image the whole brain with the high resolution very fast. So it was a dream for a long time, but our collaborator Kwang-Eun Jung in MIT developed a new tissue processing method called shield and map. Basically, to preserve the fluorescence signal and expand the tissue so we can get the high resolution imaging with the reliable fluorescence labeling. So using this approach, we injected the virus we proposed before into the globus pallidus of Pavlov-Queen animal. And then we did shield and map processing, and they could clear the brain and expand the brain while preserving the signal. A good thing about this method is while we can see the endogenous fluorescence protein, we can keep doing immunostaining and in situ hybridization, so we can actually get the labeled signal as well as cell type marker with the immunostaining and fluorescence microscope. So here is the high resolution image we acquired of the globus pallidus neuron, Pavlov positive neuron projecting to many different targets. And then we picked the one neuron, we actually did the single neuron tracing and traced the gray line is actually the axon fiber traced kind of manually. And then while we are doing, actually, we are able to do in situ hybridization to actually mark the SNR neuron with many different cell type marker. So after the in situ, after the single neuron tracing, we actually get this kind of diagram. So showing that one neuron make a synapse onto the many neuron in the globus pallidus, and then axon is projected to the SNR, and then actually to make a list like synapse onto the 10 more neuron in the SNR. So this high resolution imaging of the single neuron enable us to trace single neuron level, and then we are able to get the full the organization in the long range projecting neuron in cell type specific manner. So one more step forward, because we know that there's two different populations which projecting the different target may belong to the different anatomical organization, but in our study also showed that this two neuron actually has a different electrophysiological property. So looks like they are different anatomically and functionally. So we thought that they may belong to the different brain-wide circuitry, right? So we actually used the other viral tool. So using the same retrograde EIAB virus expressing lipid combination dependent manner into the SNR or paravascular nucleus. And then we injected the many helper virus to label the to get the monosynaptic rabies virus infection. So inject the AAV virus expressing TVA in fluid-dependent manner into the globus pallidus of the Pavlov-Queen animal. And then later we inject the pseudotype rabies virus expressing GAP into the globus pallidus. So we are able to label the Pavlov positive neuron in globus pallidus only projecting to SNR or PF. Indeed, when we analyze the whole brain cell distribution, we found that the neuron projecting to PB neuron in the globus pallidus are actually different based on where those globus pallidus neuron project to. So overall, I provide many different viral tools. And And then this is a short summary that using viral tool, we're able to differentiate the axon fiber and synaptic bouton. I think that it's very important to fully understand the anatomical organization of the specific neuron. And then using a novel tissue processing method, SHIELD and MAP, together with the single neuron tracing, we are able to get the high resolution single neuron imaging, single neuron processing in the most reliable manner. So I don't have an ethnology slide here, but this work is done by collaboration with the Pang Wen-Jiang, as I mentioned, and then also the my lab members in UCSD. Yeah.
Thank you for that outstanding presentation. For additional panels on the NIH Brain Initiative, a multidisciplinary approach to neuroscience, please view our agenda on our events page. As a final reminder, the panelists will follow up with any questions you've submitted via email. Thank you again for your participation.